In this section, we're going to focus on examining energetics of life and how we get energy out of our environment. Really, all of the energy in our bodies ultimately comes from energy that was stored in the sun. The sun is burning different gases and it releases electromagnetic radiation that is then taken up by plants to undergo photosynthesis. So photosynthesis is basically taking small sugars or carbon dioxide and turning it into sugar. And those sugars can then get converted into longer polymers of sugar that can be starch or cellulose. That is then consumed by animals like us, and they also then break down that stored energy as long chains of sugar, and they can do work that can generate heat, can maintain body temperature from brown fat. So there are different ways that this process works. We're going to talk about how we convert this energy into something that we can utilize and use. There's a few necessary components of what we consider energy or food that's going to work properly for our bodies and our systems. One of the things that we need to have is that the food that we consume has to release that energy gradually, where we can't just have an explosion of energy in our bodies. It has to be released over a period of time so that we can harness that and utilize that effectively. It has to be in some type of accessible form. So it has to be something that our body can recognize and utilize consistently. We have to be able to control when and where we use that energy. And we also need some heat for our body temperature, because we talked about enzymes, right? I know that when you stick your hand in ice cold water, it's really hard to use your hand. And that is the reason we need heat for our bodies, so that our enzymes can effectively function at the speed and the rate that we need them to. Energy also has to be available to drive different types of chemical reactions. So it has to be an accessible form, but also has to be consistent and consistently available to run the mechanisms within our cells. We're going to talk about thermodynamics. So if you've taken other chemistry courses, this has likely come up a lot, but it's important to think about the balance of energy and the balance of reactants versus products. And so we're going to talk about this process of how we take carbohydrates, fats, and proteins, utilize those for energy, and some of the products that we're making. So we have to think about the equilibrium and where things are coming and going so that we can understand what processes are going to function normally, and if we inhibit any of those processes, what then pathways are available for that equilibrium to shift towards. Equilibrium and of thermodynamics really helps to explain what happens if we have a lack of oxygen, or if we have a buildup of carbon dioxide in our systems. This theme is really important because there are millions of chemical equations that are running simultaneously in our bodies, and the reaction rates and the equilibrium of those rates are constantly changing. So this class, we're really focusing on the various mechanisms of our cells and how they regulate these rates and utilize different chemical equilibria to drive those different reactions, and how inhibitors and activators can change those dynamics. We've already discussed reaction rates, but I'm going to quickly go over this again just as a review. For a generic reaction, A plus B turns into C plus D. The rate of the reaction is related to the concentrations of the reactants. So A times B, and this would be a second order reaction, right? We're assuming that both of those things plays a role in the speed. So that's generally the case in biochemistry. Well, the amount of reactants we have is going to dictate how fast that goes. And for any kind of reaction, each of those different components, A and B, we can be an infinite number of different things coming together, but the certain different types of things will have its own constant. So it has a rate constant that's defined and determined by the concentrations of these reactants. So just an example of this, if we lose their ability to breathe and we don't have oxygen available, this chemical reaction cannot move forward. Hydrogen ions that are created and generated and enriched within the mitochondria of our cells, those are not going to then be able to chemically react and resolve this chemical reaction, and we're not going to be able to make ATP, and that's going to backlog other processes and death will occur. So in chemistry, we talk about A plus B will generate C plus D, right? So we have one reaction constant, and the reverse reaction would have that inverse reaction rate constant. So most biochemical processes are reversible. However, each step is regulated by an enzyme, and sometimes enzymes don't catalyze the reverse reaction very well. We talked about enzyme kinetics and how the product of that substrate generally has a lower affinity for that enzyme pocket than it does for the reactant. So the reverse reaction sometimes can work. It depends on the enzyme, but it doesn't always work. Sometimes there's a much lower affinity for the reactants than the products, and that will determine if that chemical reaction can go backwards or not. At equilibrium, the rate of the forward reaction is equal to the rate of the reverse reaction. So this is what we see at equilibrium. If we take our K1 over K negative 1, that is going to give us our equilibrium constant. Here's an example of ethanol and oxygen that we're going to generate this aldehyde and water. And you can see here that the rate of the reaction, these are the reactants and the products over reactants always. If we look at the term, you'll notice that the H2O, we have this is to the second power because there's a 2 in the coefficient. And so we have that same here for the aldehyde. The oxygen has just a 1 coefficient, so it's to the power of 1, and then we have a 2 coefficient, power of 2. So for other chemistry classes, this has been covered. We're not going to go into too much detail in here, but I just wanted to cover this quickly. So for example, let's pretend we have 4 moles of water, 4 moles of this aldehyde, half a mole of oxygen, and 1 mole of ethanol. So that would equal 128 mole. That would be our equilibrium constant, 128 molar. So if we remove some water and make this instead of 4 moles of water, we're going to then now make 1 mole of water. So we're going to put 1 mole of water here. So we have 1 squared, 4 squared, 0.5, and 1 squared. Again, that's going to give us 8 molar. So what do we need to do to increase this value back to 128? So we've taken some of this product out, and that changes this KEQ, which is it's now actually not at equilibrium. It's out of equilibrium. So this will push back towards that equilibrium value. So in order to get this back to the original 128 value, we need to increase the numerator, or the products, or decrease the denominator, or the reactants, to increase that value back up. So that's really what we think about. That's called Le Chatelier's principle. So this is Le Chatelier's principle. We're thinking about any change that's imposed on the system. So if we change the amount of reactants or the products, if we pull D out to go through the citric acid cycle, this is going to cause a shift to replace that and reestablish that equilibrium based on this idea of products over reactants. So these concentrations will want to go back to those relative ratios. Metabolic pathways are series of reactions. So each of these nodes in here, you can see each of these little nodes is a different organic compound that's being processed. And each of these arrows are being mediated by different enzymes with very specific functions to enzymatically process that specific compound. So we have a ton of these chemical reactions happening at one time, and we have a lot of different things that are pulling on this process to change consistently and constantly that equilibrium reaction. So these chemical reactions are hardly happening in a test tube. They're happening in this dynamic system that's constantly changing due to a lot of different factors, right? How fast this process is pulling from here, how fast this process is dumping into that process. So all of these things are relying on each other. There are different forms of metabolic pathways. So there's a linear form that's shown here. We just have one reactant turns into another, turns into another, turns into another, and it's just kind of that's the way that it goes in one direction. There's a cyclic version as well, where we kind of add things and change things throughout this process, but we kind of have this creation of something that then gets formed into the starting point material, and we start all over again. So there's a cyclic here, and then there's spiral assay as well, metabolic pathway, where we kind of slowly chip away at a certain molecule. So this is what happens with fat, either synthesis, we're going this way, we're making it bigger, or fat catabolism, we're actually breaking it down. I have introduced you to inhibition already, and there will be different types of inhibitors. And as we start thinking about inhibitors, not just for one chemical reaction, we think about where these are coming from. So a lot of times in our metabolic processes, when we generate some type of product, a lot of times when there's a lot of that product, it can feed back and act as an inhibitor to one of the earlier steps in the reaction. And this helps to make sure that we don't generate too much of this product. When we have sufficient amounts, it'll inhibit this process, and then A can go on to one of the other many processes in our bodies that we might need that compound for as well. In addition to having inhibitors, there are things that are feed-forward activators as well. So we can have activators that help to increase the efficiency of the enzyme. We can think of those allosteric activators. And so this is something that feeds forward. If we generate a lot of B, that might feed forward and help to pull the equilibrium towards the products to help to reduce the amount of B so that we can generate our products. So different ways of regulating these different metabolic processes. Okay, so we can predict the rate and direction, but what about energy? Gibbs free energy helps us to describe the energy of a system and if that chemical reaction is going to move forward or not, and how much of that energy is it helps to describe the change in energy from the reactants to the products. We're going to start with the Gibbs free energy. And we're also going to have within this amount, we're going to look at the delta H, which is the enthalpy. So the change in the enthalpy from the change from the reactants to the product, minus the temperature in kelvins. And also we're going to look at the change in entropy. I'm going to explain what entropy is in a little bit here, but we have usually Gibbs free energy is given in kilojoules. And if it's going to happen, if the chemical reaction is going to happen, this is generally speaking, right? This is everything's based on probabilities, but in general, if something has a positive free energy, a change in free energy, it's not spontaneous. It's not going to generally happen. So it's also referred to as endergonic. If it's a negative value, it's more likely to occur or to be spontaneous, and that's called exergonic. The change in the enthalpy is going to be in kilojoules generally also. We've heard these terms before, endothermic and exothermic. We're talking about kind of the heat energy that's involved. The temperature is always going to be in Kelvin, and the reason for that is Kelvin values are never negative. So the temperature is always in Kelvin. That kind of helps us not mess around with positives and negatives. And then the entropy value is going to be in kilojoules over Kelvin degree, right? Kelvin. And this helps us, then we can subtract, right? When we multiply these together, we're going to get a kilojoule value that we can subtract from this kilojoule value of the enthalpy, and then we'll have the ending units be the same. So this is kind of what we're going to be looking at and thinking about in the next few slides here. This is an example I'd like you to be able to walk through, and we're basically looking at the changes between the reactants and the products. So you have to identify the reactants here. So we have that ammonium nitrate is a reactant. These other two are products, and you'll notice that water is missing. And since we're in an aqueous environment, we don't count water as part of that reaction equation. And in order to figure this out, to get the delta H value, right? We know that Gibbs free energy is delta G is equal to delta H minus temperature times delta S. And so in order to kind of calculate this, we want to first calculate the delta H value, then we want to calculate the delta S value, and we know the degrees in Kelvin I've given you on the top there is 298.15 Kelvin. We can calculate Gibbs free energy. So let's start with how do we figure out the delta H value? We have to take the product values and subtract them from the reactant values. The delta H is going to be, and we have just 1 mole of all of these. There's no prefixes here, so they're all just 1 mole of these. Look at the units as kilojoules per mole. So we're going to have to take, so for NH4 we have 1 mole of this, and we're going to multiply that mole times the negative 132.15 kilojoules per mole. We're then going to add to that the 1 mole of the nitrate. That group is going to be subtracted, the products minus the reactants. That's going to be the 1 mole of the ammonium nitrate times the negative 36556 kilojoules, and apologize for going up here on the side. That should, in the end, the delta H then, if we calculate that, use a calculator, should give us something like 28.05 kilojoules. 28.05 kilojoules for delta H. And now we have to do the same thing for the delta S value. And this time I'm going to start over here so I don't run off the page. And for the delta S, we have again the products, and we're going to subtract that from the reactants. Same thing, we have 1 mole times 113.4. Now we have joules, not kilojoules, per mole Kelvin. We're going to add to that the 1 mole, 0.4 joules per mole Kelvin. And we're going to subtract that, right? We're getting the difference. It's the delta value of the 1 mole, 151.08 joules per, should then equal 108 joules per Kelvin. We have the delta H value, we have the delta S value, and if we're also next going to determine the delta G value, which is what I want to do next, I am going to then take these values, right? I'm going to take the T value, which is 298.15 Kelvin. I'm going to multiply that with my 108.7 joules per Kelvin. And I'm going to take my delta H value. I'm going to convert this from kilojoules to joules. And our final delta G then should equal negative 4,360. Since this is a negative value, we would expect this to be a spontaneous reaction. Now what I'd like you to try is to recalculate this if the temperature is negative 20 degrees Celsius. So let's look at this Gibbs free energy equation. And we're going to now, I would like to have you examine and look at the differences between what these actually mean. So if we have, again, that negative value here, if it's negative, if the delta G is negative, it is going to be a spontaneous reaction. If it's positive, it's most likely not going to be spontaneous, it's not going to move forward. So that means that if these are positive values, something has to happen with the system to change it. We have to add energy, we have to do something. A lot of times in biochemistry, we actually couple reactions. So we'll have two reactions go together. If the one reaction that we want to have move forward is endergonic, if we couple it with an exergonic reaction, we add, for example, we can break bonds in ATP. That is an exergonic reaction. ATP bonds like to break. We can add that energy to this whole system to make things that normally wouldn't move forward. So that's another trick of biochemistry. In addition to using enzymes to make things go faster, we can actually couple reactions that normally allow things that normally wouldn't happen to happen. The idea here, this idea that we want this to be a negative value, positive changes in the entropy are going to reduce this value, right? Because it's a more negative value then. And that's going to promote giving spontaneous reactions. We're going to want to create more disorder, right? We're going to want to create more disorder. And what does that mean? Let's look at what entropy means. Disorder is this idea where disorder and extra heat lead to spontaneous reaction. It doesn't say anything about how fast it's going to happen. It's just what is the probability that a chemical reaction is going to happen. So this is kind of the idea I brought up. I'd like to bring up is where your room, your dorm room or your bedroom at home, you have basically it's natural for that to become messy. Over the course of time, your room is a natural spontaneous reaction. Messes happen. It's harder to clean up, but in order to reorganize everything, in order to put everything back into order, right? This is disorder right now. In order to put everything back into order, you have to put energy back into the system. You have to spend energy to put all of those shoes away, do your laundry, fold up your clothes. It's too bad that dishwashing isn't a spontaneous reaction, but we know it's not. If we look at this on a cellular level, now going from your bedroom to your cells in your body, this is something that your cells are doing all the time. They're putting tremendous amount of energy in putting everything where it should go in your cell. It's constantly spending energy to compartmentalize, localize various proteins, lipids, amino acids, DNA, right? All of these little organelles here are little compartments of things that are supposed to be organized and put together. And that all takes energy because we're ordering things, right? So whenever we make order, that causes us to spend some energy, or that energy has to come from somewhere. Whenever we get DNA in this little box here, in this little bag here, it has to all be ordered, and it takes energy. So I want you to be able to think about this in when we're talking about metabolism, when we're talking about breaking things down, right? When we break order, when we break chains of molecules, that should release energy. That should give us energy. When we have to create big, long chains, that actually creates order. We're organizing things when we're making big chains. And so this should be an idea in your mind that starts to make sense. And it's now described by this Gibbs-free energy law. So it's kind of interesting. We can actually connect this with the real world and what we kind of in nature. This is just an example of compartmentalization, even in bacteria, right? We know these are single cells, but you can kind of see that even bacteria can create these little rounded cells. We can see these little rounded cells at the end. They're heterocysts, and they form specialized, they're really specialized in fixing nitrogen in the air. And it's a lot of nitrogen gas in the air. It's one of the very few bacteria, one of the few things on Earth that can actually pull nitrogen out of the air. So it's really important, right? We know nitrogen is an important component of amino acids and proteins. If we didn't have these bacteria on our planet, we wouldn't have much life going on because we wouldn't have enough nitrogen source to sustain it. So another kind of way that we can, from disorder, from something, nitrogen gas is really stable. N2 is extremely stable. These bacteria are able to break that apart and make other things and harness that energy. So these two terms that we'll talk about here is catabolism, and that's breaking down food, and anabolism is building up larger molecules. And whenever we're catabolizing something, whenever we're breaking something down, then we are increasing our entropy. That delta S value is going up. And when we take smaller molecules and build larger molecules, that delta S value is going to be negative. So this process can release energy, and this process requires energy. So that's the simple diagram is describing that. This is a very important concept in metabolism. Endergonic reactions, these are non-spontaneous reactions. Endergonic, right? So it's like endothermic. It's a similar prefix to that word. Non-spontaneous reactions or processes that require or absorb energy, and it will have a positive delta G value. An example of this is photosynthesis. So when we take carbon dioxide and water, right, we have plants that have chlorophyll. They can absorb light, and they can take carbon dioxide and water and make sugar molecules. So this actually requires 686 kilocalories per mole. That's endergonic. It requires energy. When we eat that sugar, we breathe in oxygen. We break that down. That's an oxidative process. And that actually releases 686 kilocalories per mole of energy. Or that energy is maybe not released as heat, right? We could do a lot of different things. And so that exergonic reaction, that's where the delta G value is negative. So it depends on just which direction you're going in this chemical reaction, if it's going to be positive delta G or negative delta G. Now, of course, we don't just do this in one fell swoop. We go through many processes to get down to this stage of CO2, and that's what glycolysis is, the citric acid cycle, all of those things that we'll talk about here in the upcoming lectures. So plants are kind of amazing because they're really the only organism that can harness the sun within their cells. And humans have figured out other ways of doing that as well, through solar panels and stuff. But plants and humans are the only organisms that are able to figure out how to take that energy from the sun. But that energy from the sun is absolutely essential for life as we know it. All right, so you should look at this, and if we're looking at which of the following reactions would have a positive or negative delta G, you should be able to figure out, based on the recent discussion that I've had, which one of these would be positive or negative. If you're not sure, then I would rewind and kind of listen to some of the previous slides to figure that out. But I'm not going to provide the answers here right now. Similar, but a little different than that last slide, where we're not looking at delta G. If we're just looking at entropy, which of these following reactions would we have a positive or negative entropy for the change in entropy for these processes? Again, I'm not going to give the answer. If you're not sure, go back and listen to the previous slides. Otherwise, this is a good question to ask me during my office hours or class. scientists and food scientists, we can look at this, the energy that's associated with different compounds. This is a bomb calorimeter where we have an enclosed chamber. We have oxygen, plenty of oxygen to come in here in a sample cup. And if we fully burn and ignite this sample to completion, so it makes carbon dioxide and water, that will release heat in this system, and that heat energy is going to be the total delta H value. We're going to be able to calculate based on the temperature change and the heat transfer from this container to the water. So this is how we can determine the delta H values using calorimetry. And we often do this with all of the food that we eat. So we're going to start getting into looking at some nutritional values, nutritional components on food products, and looking at how those calories actually are converted. So we were talking about before joules. Joules, just a different unit. It's like the difference between grams and pounds, right? So, or kilograms and pounds. They're talking about the same thing. They're just in different units. Typically, if we're talking about food, we're talking about calories. A lot of times if we're in chemistry or looking at more chemical types of reactions, we talk about joules, but it's just a difference here. So a question I have for you to think about is, if you purchase a Happy Meal at McDonald's, what molecular components comprise these different foods? So I'd like you to choose from lipids, carbohydrates, amino acids, or just different minerals and salts. So think about cheeseburger, fries, and Coke. What do you think? Just make a hypothesis. Don't just look it up right now. Make a hypothesis as to what you think that food is made up of. What is the majority component of the cheeseburger? Does it have all three? Does it have just one? Same with the fries and the Coke. And I'll give you a second to do that, and then resume, and we'll talk about that. So these are the chemical breakdowns. I want you to start thinking about. We did some of this work looking at what happens when you eat these things. There's a substantial amount of fat in a cheeseburger, probably from the cheese and the meat that's in there. Carbohydrates, there's a lot of carbohydrates just in the breading. Proteins, 15 grams, and quite a bit of sodium as well. The calories are 300 calories for that cheeseburger. What you should start also doing is we need to start looking at saturated fat and added sugars to our diets. You want only 10% of the saturated fat in your diet to come from saturated fat, and 10% added sugars at maximum. So these are the recommendations currently from the Food and Drug Administration for dietary guidelines. Sodium also, there's an amount of sodium in there that off the top of my head I can't remember, but these are things that we should start looking at when we're eating foods. French fries, I was actually surprised about, not with the fat. I was kind of assuming there was going to be some fat because they're deep fried. Carbohydrates, I expected them because they're coming from the starches. Proteins, I was surprised that there was really any protein in fries when I had first done this. Calories, about 230 calories from that. My guess is a lot of these are, saturated fats in the cheeseburger. I'm not sure. It depends on the oil they're using for these French fries. Coke is all carbs. So if you don't get Diet Coke, you're just eating carbohydrates, and some sodium as well. Proteins are 4 kilocalories calories per gram. Fat is 9 kilocalories per gram, and carbs are 4 kilocalories per gram. So these are kind of the things that proteins and carbohydrates have a similar energy density per gram, and fat is about double that for the other two. You can also look at apple slices. Low energy density here. A lot of these, just a few carbs in here. A lot of it's fiber, so that fiber actually doesn't count towards, even though it is a carbohydrate, it doesn't count towards our calories because we can't actually consume that. It passes through our body as fiber. It's unprocessed. We'll talk about that. Don't eat the toy. It's not good for you. Plastic. Okay, so we're going to look at, again, I mentioned before the bomb calorimetry. We can do kilocalories or joules. And the idea of a calorie is that it's basically able to increase the temperature of 1 gram of water by 1 degree Celsius. So it equals about 4.2 joules. Large calorie or kilocalorie that I was just talking about for food, we use that a lot of times, the 4 kilocalories for sugars or carbohydrates and proteins, and 9 kilocalories for proteins. This is the energy increase the temperature of 1 kilogram of water by 1 degree Celsius, about 4,200 joules. So we're always, whenever you see calories in any food product, it's always actually kilocalorie. It's a big C calorie. But the small calorie, gram calories, this is typically not used in food science anyway. So we've seen these reaction coordinate diagrams before, looking at the free energy on the y-axis and the reaction progress on the x, where we have reactants starting with a certain amount of energy. They have to go through this activation energy. Once they get over this activation energy, they can fall down to the products, revert back to the reactants, but there's this intermediate state, this transition state. And the overall, right, if we go from reactants to products, if that overall energy is negative, that's also going to correspond to that Gibbs free energy being negative. These are more likely to happen. These are what we would call favorable or spontaneous reaction. Here we have a non-spontaneous, unfavorable reaction where we have this delta G is positive, the intermediate state, and then the final products have a higher delta G. When you think of this, just because something's not likely to happen or unfavorable doesn't mean that it cannot. This activation energy is a certain amount of energy that needs to be achieved by the different products. And so you can think of this as a kind of probability idea. So any amount of, the higher the temperature, right, the more energy the overall system has, the more likely it is that your products or your reactants are going to get that certain amount of energy. So you can think of this as different faces on a different die. If you have a 6-sided die, 8 or 10 or 12, the chances of you rolling a 6, let's say that this energy, this activation energy, is 6 arbitrary units. And to get that, right, there's only one chance in 6 for a 6-sided die. But if you go to 8-sided die, right, you could roll an 8, a 7, or a 6. So there's 3 chances out of 8. That's 37%. When you go 10-sided die, it's 50% chance. 12-sided die is 58% chance. So you can kind of think of this idea, the more we increase the temperature, the more likely that your reactants are going to have enough energy to jump over this activation energy threshold. All of these following things, the concentration of the products, the temperature of the reaction, the pressure of the reaction, concentration of the reactants, they're all going to influence the chances that these molecules are going to meet and react. And these are just standard conditions for where pH of 7, mole for every reactant, 25 degrees Celsius, 1 atmosphere of pressure, right? These standard conditions are for in a chemistry lab. We know that in biochemical conditions might change depending on the type of organism you're working with or where within that organism these things are happening. But it'll influence those reaction probabilities. I want you to take a few seconds to look at this question. You should be able to answer this. If you can't, go back and look at those other slides or listen to the other slide descriptions. Biochemistry is the combination of a lot of different energies, a lot of different chemical reactions. And these are state functions, which means that it doesn't matter how you get from point A to point B or point B back to point A, right? So you can go through 550 different chemical reactions. That amount of energy that has to go in versus that comes out when you go back down is going to be the same. So in biochemistry, we don't just kind of burn off. We have glucose. We don't just have a big combustion reaction. All the energy comes out at once. We have these smaller little reactions that pull out energy, smaller bits of energy throughout the process. So one thing that we get energy out by generating ATP, which we're going to talk about in a bit. We also generate reduced coenzymes that we can use in the citric acid cycle and the electron transport chain. So if we don't need that energy right away, that's one of the benefits of biochemistry, is that we can take these little bits of energy and save them for when we need them. And so one of these things that we do is we actually generate these triphosphate bonds, adenosine triphosphate, ATP. Right? What are we doing? We're actually building order. We're making longer, bigger things. So in order to make something bigger, we actually can create this longer chain. And that gives us a little bit of that we, more order, less disorder. So how we can use this as quick money transfer, energy transfer in our system. So it's energy transporter, this ATP. It's how things change hands. And again, the $100 bill is maybe not the best example. Might be a $20 bill that's a better example, because more people have $20 bills in everyday life, and so that's more of a common amount of money that might change hands, or you might use in a grocery store, for example, than a $100 bill. So it's something that's really easily accessible. We can actually see here in a second, we'll actually look at here that ATP, if you look at the types of free energies involved in the generation of ATP from ADP, we have negative 32 kilojoules per mole. That's intermediate here, right? Generation of phosphoenol pyruvate, or ATP to AMP, we get different amounts of energy, and that's an intermediate state. So it's not the largest amount. We always think of the ATP, it's a high energy bond. Well, it's not really that high of an energy bond where we really look at it. It's an intermediate energy bond that is easily manipulated and utilized in our cells. And it's oftentimes used to couple reactions. So if we have ATP present, we can break that bond, and we can use that as a part of an exergonic reaction. So here's a favorable reaction that's going to be coupled with a non-favorable reaction. And these products might, form these intermediate reactants, and then this overall exergonic reaction drives it forward, and so it pulls this whole process forward. So the coupling of reactions helps to couple these things together. So this overall reaction might have a negative delta G. So let me give you an example of this. So normally glutamate has carboxylic acid side chain. It's an amino acid. If we were to add ammonium ion to that, the formation of this would generate glutamine and water. And this overall reaction is positive, it has a positive delta G. So it's not going to likely happen on its own. It's not a favorable reaction. However, if we look at this, ATP, the hydrolysis of ATP into ADP, we get negative 32 kilojoules. So this is a very favorable reaction. The breaking of this bond to ADP is favorable. So how can we combine these things? Here's glutamate, that compound I just was talking about. I mentioned it had a carboxylic acid. ATP to ADP is actually going to transfer one of these phosphate groups to that carboxylic acid side chain here. And then we have this intermediate step. And now we're going to bring in ammonium, and it's going to pop off that phosphate group. So we're going to get inorganic phosphate. And now we have an amide functional group, which changes this from a glutamate to a glutamine. And that's how we can actually make this favorable. So now the overall, when that step one is going to be negative 32 kilojoules per mole. And step two here is going to be plus 14 kilojoules per mole. The overall delta G, the net, when you add negative 32 and 14, is going to be a negative 18 kilojoules per mole. So now this became spontaneous. This became a spontaneous reaction by coupling these two reactions together. So we're going to start looking at these processes, how we're going to take energy out of this food and break it apart, break those larger bonds down into smaller chunk-sized portions. We're going to generate heat. We're going to pull off ATP and generate chemical energy. And so that's that process we're going to start here. So I'm going to stop at this point, and then I will continue. After we have a case study, we'll continue on the introduction to human metabolism and some of the important metabolic processes we'll be talking about for the rest of this semester.